Now, what are the enzymes? Enzymes are the biological catalysts are produced in the living cells which catalyze the so many biological reactions in the living organisms. That means enzymes act as a catalyst for the so many biological reactions. Take the energy profile diagram for any biological reactions. In the absence of the enzyme, this is the energy variance of the biological reaction. But in presence of enzyme, this enzyme decreases the activation energy of the biological reaction. Hence, the reaction gets faster. Take a simple example. Without having any enzyme to digest the single plate meals, it will take 50 hours. But in the presence of enzymes, this will happen in the 1 hour or like that. So, due to these reasons, enzymes are the more essential components in our body. Not only in our body, all living systems. And these enzymes have a colloidal in nature and get inactivated during the reaction. These are colloidal in nature and get inactivated during the reaction and have to be constantly replaced by the synthesis in our body. That means after getting the reaction, it is to be inactivated and again it is reused by the biological reactions. Now take a simple example, hydrolysis of sucrose. In this case, invertase is an enzyme. And sucrose, when it comes on the surface of the invertase and it is adjusted according to that, and the hydrolysis is occurred and it gets the fructose and glucose as a products. In this case, invertase act as a enzyme. So, in the presence of invertase, this sucrose gets hydrolyzed to fructose and glucose. Here, invertase. We already discussed that these enzymes are decreases the activation energy of the reaction and so that reaction goes fast. It forms glucose and fructose as a product. Now you can see the another examples in the table, that is urease in an enzyme, and it is used to convert the urea to carbon dioxide and ammonia. This urease is used in the conversion of urea to carbon dioxide plus ammonia. And invertase, we have already discussed it. Sucrose can convert to glucose plus fructose in presence of invertase. And maltase in enzyme which converts the maltose to glucose, 2 moles of glucose. And lactase, this lactase is useful to lactose, hydrolysis of lactose, and it forms 1 mole of galactose and 1 mole of glucose. And amylase, amylase is another enzyme which is useful in the hydrolysis of the starch. The starch is hydrolyzed to alpha glucose. And carbonic anhydrase, this carbonic anhydrase is useful in carbonic acid is converted to carbon dioxide and water. This carbonic anhydrase is very useful to maintain the pH in our body. And pepsin and trypsin, these two are useful to the hydrolysis of the proteins. Proteins in presence of trypsin or trypsin as enzymes gets fully hydrolyzed and forms the amino acids. Nuclease. This nuclease is very useful reagent to convert the DNA or RNA to its primary units, that is nucleotides. In presence of nuclease as an enzyme, DNA and RNA is converted to nucleotides. Already you have know that DNA and RNA are the primary structures or polymers of the nucleotides. In presence of nuclease and an enzyme, it's converted to its nucleotides.